J. Craig Venter has been a very controversial figure in the field of genomics, and he's done a lot of audacious and innovative things. A couple of them that I'm going to tell you about now. In 2010, he took a bacterium and took out the natural genome, all of its natural DNA, and replaced it with a completely synthetic genome that had been, completely artificially synthesized in the lab, and it worked. So this, in some sense, was a synthetic creature. I mean, he didn't start from scratch. I mean, he started from the shell of the bacteria that was there with the organelles in place and the cell membrane and the cell wall. So he didn't replace those, but he did replace the genome, and it worked. This cell with the synthetic genome could reproduce. It could grow. It could do all the stuff that the natural version of this bacteria could do. So now, this year, in 2016, he's taken this a step further, and he's taken that original synthetic genome, and he's pared it down to just the genes that are essential for the basic functions of life. So we're talking about a bacteria here. We're talking about basically just assimilating nutrients, growing, and then reproducing, by cell fission, the way bacteria reproduce. And he's pared it down to what he calls a hypothetical minimum genome. And it is actually, he developed it through a very rigorous process of finding out which genes are essential for just the basic functions of life. Now, the cell doesn't do anything other than that. And so, and in fact, it can't live outside the laboratory because it can only grow if you feed it all the nutrients it needs. But this is called he calls this JCVI, that's for the J. Craig Venter Institute, JCVI Syn 3.0. It's the third version, or the 3.0 version of this bacterium that they've come up with, and it has 473 genes, and that's it. Now, just to put it in, and this cell can do all those things. It can assimilate nutrients, it can grow. When it's grown enough, it can, divide into two and thereby reproduce. Now, the synthetic genome, this minimum genome cell, is very interesting. Just to put it in perspective, if you look at some of the bacteria that have been studied a lot, for instance, like E. coli, it has a genome of about 5,000 genes, give or take. Humans have a genome of about 25,000 genes. So, but it's of interest to note, what is the minimum number of genes and what they do just for the basic stuff of a living cell. They have a long list of the genes that they've retained, and of course they have a long list of the genes that they've gotten rid of. And I want to show you a pie chart of that, and I think maybe it tells us something about living organisms and what it takes for something to be alive.
Okay, the article that described this minimal genome was published just a couple of weeks ago on March 25th. It was published in Science. It's an article by Hutchinson and all these other authors. You can see the senior author is J. Craig Venter, who I mentioned previously. One thing I think is really interesting is the list of the genes that they retained, that they found to be essential for the basic functions of the cell. One thing I thought was really extremely interesting is that 41% of them were for gene expression. And they were about, transcribing DNA to messenger RNA, making ribosomes and all of the raw materials needed for protein synthesis. Only 7% of the synthetic genome was used for DNA replication and cell division, which I thought was sort of interesting because DNA replication and cell division are so important, but evidently gene expression is a bigger piece here. 18% of the synthetic genome was for maintaining the cell membrane, including membrane-bound proteins. And then 17% was for metabolism that we talked about a couple of weeks ago. And one other fascinating thing is that 17% of the genes have no known function. Yet, Venter and colleagues found that the 17% of the genes were necessary for the cell to live or for it to thrive. So that's pretty much it.
Okay, thanks for listening.